Hi guys, this is Alicia with Good Morning Sunshine. So today we have 4 lectures and a lab. However, the lectures, we have 6 PowerPoints to get through, so it's going to be a long one today. 4 of them being anatomy, and then we got into muscle physiology. So starting with general concepts regarding limb muscles. We want to recall that muscles are often arranged in layers. So they usually have a superficial layer, and then it goes to a middle layer and then to a deep layer. Also recall that muscles can only shorten, thus they can only ever pull, they cannot push. And muscles act only on joints and not on bones or the limb regions themselves. So basically, like the triceps brachii extends the elbow, not the humerus or the antebrachium. So they only act on joints, not muscles. Sorry, muscles only act on joints, not bones themselves. So 2 exceptions actually to this rule, of course there's always exceptions, would be the carpus and our digits. So carpus being our wrist and digits being our toes. If a muscle crosses a joint, it will act upon that joint. That is a good concept. This includes actions on multiple joints in many instances. We also have the concept of major and minor, or primary and secondary actions. So the major one is usually with the bigger muscle acting on, and then the smaller muscle is usually our minor muscle. And then we have heads versus bellies of a muscle. So that's the naming of multiple parts. Some muscles are composed of a single mass, and then we have other muscles that are composed of multiple chunks. So they would have different heads and maybe different bellies as well. So here we have the supraspinatus, that's just one big muscle. But then we also have the triceps brachii, which has multiple heads as well as multiple bodies. If all parts or chunks insert in the same place, it's just called one muscle, just like the triceps brachii. Even though it's subdivided into like 4 different bellies, it's still called just the triceps brachii, but it has different chunks. So it has a lateral part, it has an accessory part, it has a medial part, and it has a long part. But all of them are one common muscle, which is the triceps brachii or brachii. So each chunk is considered a head. So example, the triceps brachii is comprised of 4 chunks, just like I said. All 4 heads have separate proximal attachments, but converge on a single tendon to insert at the same place. So they all insert at the olecranon tuberosity, which is basically our elbow here, if you can see in that picture. So a head of a muscle is an individual chunk of a single muscle that originates from one particular place. Just like I said, the olecranon tuberosity for the triceps brachii. Sometimes a single head is itself divided into separate parts. Then each of those bits is a belly. So chunks can have different subchunks basically, and these subdivisions are called the bellies. The belly is multiple parts of one head. And then we have the bursa or bursae. This is our friction reducer, a synovial balloon. It's placed between 2 high pressure points. It's often between a tendon and a bony prominence, and it rolls between the 2 structures. So there's no like scraping in between. And also it reduces friction and allows the muscle to roll. And then we have tendon sheaths. The long tendons. Long tendons don't heal very well. It's running right over the bones, would be vulnerable to friction injury. So tendon sheaths are synovial structures, kind of like a double over bursa that protect the tendons from such damage. And then we went over stuff in lab today. We skipped a lot of things, so I'll be skipping around. We have extrinsic muscles of the thoracic limbs. So these muscles attach the limb to the axial skeleton. They also move the limb relative to the trunk or vice versa. They have origins, they have insertion points, innervations, and actions. And then we have intrinsic muscles of limbs. So these all attachments lie on the bones of the limb itself, thus move regions of limb relative to each other, and they also have origins, innervations, actions, and insertions. And then we have flexor angles of the limbs which define actions of major muscle groups. So example would be all muscles moving the shoulder. Extensors are cranial to the scapular spine, whereas flexors are caudal to the scapular spine. So here we have our flexor angles. Flexor here. The only exception is the elbow, which is cranially. All the other ones are caudally here, all the flexors. And then the extensors of the shoulder are on the cranial side of the scapula. Major functional groups are usually innervated by the same nerve. So example would be extensors of the elbow, all inverted by the radial nerve. This characteristic gives rise to the concept of nerve muscle joints. So our elbow extensors, again, are innervated by our radial nerves. And then we got into the intrinsic muscles of the canine brachial region. Again, we went over a lot of this in lab. The muscles related mainly to the brachium. These muscles are related primarily to the shoulder. So we have lateral to the scapula that we're going to start with. So generally, they flex or extend the shoulder joint. We have the deltoideus, which is in two parts. We have the spinatus part and then the acromial part. And then we have the supraspinatus and then the infraspinatus and the teres minor here. These are all lateral to the scapula. So the deltoideus is up here. This is the blue and green region. And then we have the teres minor here. And then we have the supraspinatus, which is, of course, the top one because supra means the top. And then infraspinatus is the purple, which means the bottom. And then we have our teres minor, which is that little green muscle down there. And then we have medial to the scapular. So these generally stabilize, adduct, and rotate the shoulder. So here we have the subscapularis and the teres major. The teres major also flexes the shoulder joint. And then we have the coracobrachialis. So to show you here, in green is our subscapularis. In blue here, this is our teres major. And then we have the coracobrachialis, which is purple down here. It looks like a little nugget. And then cranial to the humerus. These generally flex the elbow. And here we have, in green, we have the brachialis here, but then we have the biceps brachii. The brachioradialis, in dogs, it's constant, and then we have a thin slip of muscle often adhere to the skin. So it's like paper-like, the brachioradialis, but it looks like that. However, in cats, the brachioradialis is robust and it's constant, so it's actually really big in cats, and it's always there. Sometimes in dogs it's not. Cranial to the humerus, we have the biceps brachii. It's visible laterally and medially. It extends the shoulder and also supinates the paw. So here in blue here, this is our biceps brachii. Here's a better picture actually. This is our biceps brachii here. And then we have the brachialis, also known as the twisty muscle. It begins medially and proximally. It courses laterally and ends up cranially. It's mostly visible from the lateral side though, however. So the brachialis is that little piece blue there, but it's a very twisty muscle. Here's a better picture as well. So we have the brachialis here, and then we have the brachialis there too as well. Caudal to the humerus, they generally extend the elbow, these muscles. We have the triceps brachii. It has 4 heads, like I said. It has 4 heads in a dog. It's called triceps because tri usually means 3, but that's because it was named in humans first, but in dogs there are 4 heads. It's variable in a cat. And then we also have the onconius and the tensor fasciae antebrachii. These are all caudal to the humerus. So laterally, we have the triceps brachii with the 4 heads in the dog, and then we have the onconius here. So here, so again, 4 heads. So we have the triceps brachii long head right there. We have the lateral head here. And then we also have the onconius here. This is all from the lateral side, but caudal to the humerus. And then here's actually all 4 heads. So we have the accessory, the medial head here, and then we have the long head here, and then the lateral head is right there. It's just cut off. Medially, we again still have the triceps brachii, but then we have the tensor fasciae antebrachia as well, which is the big arrow showing you right there. And then the long head of the triceps, it originates from caudal scapular border, so it crosses the shoulder as well. So its action there. And then we got into the next PowerPoint. Sorry, there's going to be a lot of PowerPoint. So now we're getting into the muscles of the antebrachium and the manus. We want to look at what side the limb they're on so we can orient ourselves. Is it laterally or medially? She doesn't want us to memorize them in order because then we're just memorizing. We're not actually knowing like the muscle itself. So due to the configuration from the carpus on down, the flexor surfaces of all joints lie palmarly. So any muscle crossing the same side of any of these joints will have similar action. So we can simplify and say flexors and extensors of the carpus and digits, and that's how we would name them. When we are considering attachments of the antebrachial muscles, they show that any muscle that flexes the digits also flexes the carpus, but not all carpal flexors also flex the digits. The general naming of muscles, all muscles are named including an action and insertion region, also known as a distal attachment and a modifier. If we're saying muscles in English terms, we go from the modifier to the insertion to the action. So example of this would be lateral would be our modifier. So what side is on lateral medial. Our insertion would be digital here, and then the action would be extensor or flexor, and this would be an extensor. However, you can also use Latin in order to name muscles. In Latin, it goes from the action to the insertion to the modifier. So like I said, in English, it would be known as the lateral digital extensor. However, in Latin, it would be known as extensor digitorum lateralis. I mean, it sounds nicer, but I'm probably going to learn them in English. Anyways. So functional groupings, extensors of the carpus and digits are craniolaterally. The common origin is lateral extensor epicondyle of the humerus. The list we need to know is this one, which we kind of went over in lab. So we need to know the brachioradialis, which I showed you. We need to know the extensor carpi radialis, which I think we're doing these tomorrow in lab, so I can't show you these yet. And then we have the extensor digitorum communis. That's in Latin. This is our common digital extensor in English. We have our ulnaris lateralis, which is also known as our lateral ulnar muscle. Then we have our long abductor of the first digit, and then our supinator, which we'll be going over those in lab tomorrow, I believe. Flexors of the carpus, so we're on to flexors now. So flexors of the carpus and digits is caudal medially. The common origin here is the medial flexor epicondyle of the humerus. Flexors are more complex than extensors. They're the power stroke of step, so our flexion, so they include more muscles, more complex muscles, and more layers of muscles. And here's our list that we have to know for these. We have our pronator teres, our flexor carpi radialis, our flexor digitorum superficialis, our flexor carpi ulnaris, and then for our deep layer, we have to know the humeral head, the ulnar head, the radial head. This all included in the muscle of the flexor digitorum profundus. And then we have our pronator quadratus. So flexors on the superficial layer, these include our superficial digital flexor, as well as our flexor carpi ulnaris. This includes a humeral side and an ulnar head. So in our picture here, in blue is our superficial digital flexor, and then in purple we have our flexor carpi ulnaris. That's our ulnar head, but then we have the flexor carpi ulnaris, our humeral head. So there's two heads on that, but they are the same muscle. And then we have our flexors of the deep layer. So we have our deep digital flexor, which includes 3 heads. We have our humeral head, our ulnar head, and our radial head. So again, humeral head would be going to the humerus. The ulnar head, the head is on the ulna, and then the radial head is on the radius. So here's a picture of these. Again, in blue here, we have our ulnar head. In purple here, we have our humeral head. And then in green here, we have our deep digital flexor, and that's our radial head. And then we have our flexor manica, which is right here. Let's see, right there. The flexor manica is actually where it doesn't go all the way down to the digit. It splits where the superficial digital flexor stops. So functional groupings, we have our supinators and our pronators. So remember, supinator is when we turn our palm up, so like this, and then pronate is when our palm's down like that. So supinator, we only have one of those. Let's see if I can show you. So here's our supinator here. Here's our humerus to give you like where we're at. We have our lateral epicondyle and our supinator coming off our lateral epicondyle of the humerus. And then it lies cranially deeply more lateral with the carpal and digital extensors. Pronators, we have 2 of them. We have the pronator teres and the pronator quadratus. And those are next to our supinator, but above it. So here we have our pronator teres, and then here we have our pronator quadratus here. The pronator teres is cranially deeply and more medially with carpal and the digital flexors. Our pronator quadratus is caudally and very deep in the interosseous space. Muscles of the manus. There's several muscles are present, but few are large. We have limited individual use of digits or grasping. So we have all these abductors and digits. Here we have digital flexors. We have extensors as well. She only wants us to know the interosseous muscles. So there's 4 very similar muscles on the palmar surface of the metacarpal bones. So we have the proximal and sesamoid bone. We have the interosseous 3 muscle. Yeah. She skipped a lot of these, so. The cat special points you wanted to go over, the brachioradialis is well developed. Muscles bellies tend to be longer. Fibers go farther down on the tendons. Muscles of the actual manus are better developed, more use of the hands. And they have specialized distal phalanges, so the retractable claws. And then into our third PowerPoint, we're going over vessels and nerves of the thoracic limb. She also went fastly through this, so just bear with me. General concepts of the lymphatic structures. As everywhere, networks of tiny vessels begin blindly and coalesce to lead to the systemic circulation. Larger ones sometimes follow veins, and vessels coalesce and pass through regional before reaching large veins. So regardless, you'll never see the vessels, though you will see the major nodes of the vessels. So we have prescapular lymph nodes. So And then we have axillary, and then we have cubital, which is mainly in horses. So I can show you those. So we have the prescapular lymph nodes here, and then the axillary back here. So in the armpit, basically, of the dog. So blood vascular structures, we have a main channel flow. So we have to know the sequence of this. But the main channel flow is a continuous channel of larger vessels passing proximal to distal. So the main channel flow is like an interstate, and then you get many exits. The main channel flow, so branches from the main channel supply local regions. So these are our exits. So you have the main interstate, and then you have exits on that. The main channel really is a single tube, but it changes names in various regions to reflect the name of the region in which it lies. Since is major conduit, it bleeds the most when you cut it. And it's more on the medial side of the limb, so it is protected, so it's less likely to get cut that way. Artery general concept, so larger arteries tend to lie medially and deeply, so they're protected as well. Larger arteries tend to lie medially and deeply, like I said, but the accessory one lies laterally. The names of arteries often reflect placement in the limb and the structure it supplies. Origins or branches tend to, but do not rely on it. So we have to start somewhere, she said. So we have the main channel going through, and then we have all the exits. So we're going to have to know all of those at some point. Some point soon, probably. Regional flow, this is simply the local vessels supplying the smaller areas. So these are our exits. The red being the main channel flow here, which you see in red, and then the blacks are our regional flow areas of blood. Collateral or recurrent, supplemental stand-ins, extra flow. So these are our helper outer guys. When we see recurrent or collateral, that means they're helping out. So veins, general concepts, there's two sets of veins. We have our deep satellite veins. They're often doubled and take the same name. And then we have drain, which do not supply areas supplied by companion arteries. So portal veins only supply, everything else drains. We have our superficial veins, which are really just veins in the subacute area. And then the major superficial veins of the thoracic limb, these veins run on their own, not with similarly named arteries. So like our cephalic vein, there's not a cephalic artery, if that makes sense. We have to know where the accessory cephalic is, so that begins dorsally. We have to know where the cephalic begins, and that's palmarly. And then we have our medium cuboidal, which connects the cephalic and brachial veins. So here's the ones we need to know. So main arteries proximal to the manus. The main channel arterial flow as far as the mid-antebrachium is all the same in all species. So from the subclavian artery to the axillary artery to the brachial artery all the way down to the median artery, that's all the same, but then it gets cut off, and it's different in different species. So in a dog, from the median artery, you go continue through the median artery. However, in a cat, you would change to a radial artery. So here, we're looking at a dog. The bigger one would be, it would continue as the median artery, whereas in a cat, it would continue as the radial artery. So that main channel flow changes depending if it's a cat or a dog. But however, in both a canine and feline, the caudal interosseous artery is another major arterial contributor to the manus. It runs deeply between the radius and ulna, if you can see it there. Then we have arteries of paw. The main supply is palmar. We have superficial and deep palmar arches. Each lies between some other structures. We have a superficial, which is between tendons. And then we have deep, which, The palmar surface carries the bigger supply, whereas the arches mean is between two different structures. The arteries of the paw secondary supply, so this is our dorsal supply. Dorsal surface is a minor supply of blood. It's also superficial and deep sets, but again smaller than the palmar side. If you want to look at that. So we have our palmar ones, which you can see there's a lot more than we do on our dorsal side. And then we have metacarpal blood vessels, a doubled set, both dorsally and palmarly, one of each superficial and deep. The dorsal ones are indicated by the term dorsal, the palmar ones are indicated by the term palmar, and each courses from the CMC joint to the MCP joint. And of course, we have to name all of them, which it looks really scary. So, I think she skipped over these. She didn't go over any of these, so I have to get down in the PowerPoint because she skipped like 20 PowerPoints because she was running very behind. So our arteries of the digits, so each of these arteries supplies a single side of a single digit. One digit is proper rather than two digits in common. So each artery takes the same number of the digit itself. And then to distinguish these two arteries, we employ axial or abaxial. Remember, axial is closer to the axis of the manus, whereas abaxial is farther away. And axial is more medial, whereas abaxial is more lateral. So the artery on the dorsal surface on the inside to the midline of the second digit is dorsal axial proper digital artery 2. So we're gonna have to know how to name these, I think. Not too sure, but yeah. And then to the last anatomy PowerPoint. So a few thoughts on thoracic limb nerves. We have the brachial plexus. So she wants us to know this. The brachial plexus is the ultimate source of motor and sensory innervation of the thoracic limb. The formation of the brachial plexus formed from ventral branches of certain cervical and thoracic spine nerves. So the cranial nerves 6, 7, and 8, and thoracic 1 and 2. This is what makes up the brachial plexus. So here's what the brachial plexus looks like. It's this right here. So you have the brachial plexus, but then you have actually individual nerves coming off the brachial plexus. So those nerves are not called the brachial plexus, the ones that are coming off. So she wanted to get that point across. And it talked about the brachial plexus for a long time, but we also need to know, again, like the different nerves coming off. So you have the brachial plexus, but then you have individual nerves coming off. So you have the suprascapular nerve, you have the subscapular nerve, you have the craniopatellar nerves, you have the axillary nerve, you have the musculocutaneous nerve, the radial nerve, the median nerve, and the ulnar nerve. And these are not part of the brachial plexus. They are just originate from it, if that makes sense. So again, the components of the nerves of the brachial plexus have a motor component and a sensory component. Our motor component is skeletal muscles, duh, and then our sensory component are deep and superficial muscles. The motor component is a given nerve typically innervates all or part of a functional group of muscles. So our elbow flexors being our biceps brachii and brachialis. In terms of the motor component, particular significance of the radial nerve, this is the key nerve of the thoracic limb. Because to bear weight, the elbow must be held in active extension. If not, we can get radial paralysis. If we have damage to this, animals cannot bear weight, so they can't extend the elbow if there is damage to the radial nerve. That's why it's very important. Sensory component, this is our deep component to muscles, the periosteum and bone. Cutaneous skin component, we have sensory to characteristic skin regions of the skin and the sub-Q areas. It's most named nerves in the thoracic limb distal to the shoulder have a sensory component. The lateral continuation of the radial nerve, so we have a deep, which is the motor to carpal digital extensors, and then we have superficial, which is sensory to the cranial antebrachium. Nerves of the manus, major contributing nerves. We have motor, but this is our ulnar only, and then our sensories are radial, our median, and our ulnar. Dermatome equals our cutaneous zone. This is the entire skin region typically innervated by the single spinal nerve. Since most named nerves of the thoracic limb are contributed to by multiple spinal nerves, the dermatomes usually overlap. And then we have our autonomous zone. This is a skin region typically supplied by a single spinal nerve. It has clinical significance. Only a few nerves have no known cutaneous sensory component. These would include our suprascapular, our subscapular, and our thoracodorsal. The brachial plexus motor and sensory innervation to the manus simplified. Just like vessels, except only major difference is the only one set dorsally. There's only superficial ones. There's no deep ones. Nerves of the manus, so motor component ulnar only. This is our interosseous muscle and other minor muscles of the forepaw. Sensory, so we have our radial, median, and ulnar. Dorsally, we have a radial, which is the majority of the paw's dorsum, except for the lateral side of digit 5. Median, we have none, and ulnar, we have our lateral abaxial side of digit 5. Pulmonary, we have the radial has none. The median has digit 1 through the axial side of digit 4. And then ulnar, the abaxial side of 4 and all of digit 5. She didn't really go over those. She actually skipped those slides, so I just read them to you, but I don't know much about them yet. And then we're going to get into muscle physiology. We're just going over stuff that we learned in normal animal, which was a long time ago, feels like. So major features of the skeletal muscle. So principal functions are to execute body movement, to establish and maintain posture. It's accomplished by shortening and lengthening distance between points of muscle attachment to bone. Key properties of skeletal muscle. So we have contractility, which is the ability of a muscle to shorten, accompanied by mechanical force generation, so rolling movement. And then we have excitability, which is the capacity of muscle to respond to stimuli that regulates a state of contraction. And then we also have extensibility, which is the ability to passively stretch beyond a normal resting length. And then we have elasticity, which is the ability of the muscle to recoil toward original resting length after being stretched. So here's our skeletal muscle, which I kind of talked about yesterday. It's attached to bone. It's under voluntary, involuntary reflex control. They have multiple peripheral nuclei, and they have striated patterns. Overall, functional unit of a skeletal muscle includes active contractile elements, which are our muscle fibers, as well as passive elastic elements, which are our tendons that form the attachments to bone. So here's basically what it looks like. Here we are. So we have a bone here. Here's our tendon. The outside is the epimysium, and then around we have a perimysium, and then these enclosed, these little circle things, we have our endomysium and our fascicle, and then these include a muscle fiber, which is an individual muscle cell. The process of coordinated skeletal muscle function involves arrival and the detection at the muscle cell surface of an excitatory stimulus that is transduced into an intracellular signal that activates machinery capable of shortening the length of the cell, thereby bringing the points of the muscle attachment to bone closer together. Muscle relaxation or the reversal of contraction entails the removal of the extra and or intracellular stimuli to contract and reversal of events of excitation and contraction coupling. Relaxed muscles can be lengthened or stretched passively by the action of synergists. So our antagonists is when our muscles in a relaxed state, whereas our agonist is when our muscles are contracted. The arrival of an excitatory stimulus at the neuromuscular junction and its detection at the muscle cell surface. So these are all the steps it goes through, which is a lot of information. But comes in is the form of an action potential. It arrives at the terminus. There are changes to the ion channel activity. This depolarizing that allow ACH, which fuses with the plasma membrane. The ACH diffuses across the synaptic cleft, this leading to the spread of a postsynaptic potential. residual ACH diffuses away to be metabolized or transported back into the terminal, so it's getting ready for the next round of stimulation. So ACH is acetylcholine. This is signaling at the neuromuscular junction. So we get through how muscles contract, which we already learned in normal animal. The excitation contraction coupling, this is a mechanism by which spreading action potential is coupled to muscle fiber contraction. It involves the sarcolemma, which is the plasma membrane, the transverse or T tubules, which are enfoldings of the sarcolemma. And then we have the release of calcium from the sarcoplasmic reticulum, a specialized calcium reservoir within the cells that isolates calcium from contractile apparatus. The T tubules allow spreading of the action potential to pass into the depths of the muscle cell. They allow rapid and spatially coordinated calcium release from the sarcoplasmic reticulum into the presence of the thin and thick myofilaments that compose the contractile machinery. So our calcium channels that are involved in the excitation contraction coupling are our DHPR, which is our dihydropyridine receptor. This resides the sarcolemma and the T tubules. It is a voltage sensitive channel. It opens in response to membrane depolarization. It allows slow influx of calcium from the extracellular space. It physically interacts with and activates the RYR. RYR being the ryanodine receptor. This resides in the sarcoplasmic reticulum membrane. It is opened directly by the activated DHPR. This allows for the release of calcium that is stored in the reticulum into the sarcoplasma, where it can activate the contractile machinery. Once that happens, it activates machinery capable of shortening the length of the cell, thereby bringing the points of the muscle attachments to the bone closer together. So our thin myofilaments are composed of polymers of the protein actin and are anchored by Z discs, which define the boundaries of the sarcomere. And then we have our thick myofilaments, which are of myosin. They lie parallel to the two sets of thin filaments within each sarcomere. The thick and thin filaments can reversibly form a cross bridge via the myosin head. Parallel sets of the myofilaments are bundled together into myofibrils, which are multiple myofibrils are bundled together within the muscle fiber or cell. And then all in all, we have the action potential and muscle contraction after all of that happens. So sliding filaments, getting into how muscles actually contract. So calcium is induced, which induces a cross bridge formation. So when sarcoplasmic calcium is low in a resting muscle fiber and the thin filament protein tropomyosin prevents interaction between the actin and myosin filaments. So this is when our muscles are at rest. However, when the sarcoplasmic calcium increases after it's released from the sarcoplasmic reticulum, the calcium binding to the troponin promotes a change in the conformation of the tropomyosin, which then allows the myosin head to access its binding sites on the actin. So basically, myosin is now acting on the actin. And then we have a cross-bridge cycling. So we have a resting fiber, so our cross-bridge, so it's not attached to actin. And then we have our cross-bridge that binds to actin because calcium is induces this. And then we have the power stroke, which causes filaments to slide. So the power stroke involves change of orientation of the myosin head, like I talked about, which forms a cross bridge, and the dissociation of ADP and P. A new ATP binds to the myosin head, which has allowed it to be released from the actin, and then the ATP is hydrolyzed, causing the cross bridge to return to its normal orientation. And that's how muscles contract. Cross-bridge cycling, so overall shortening of the myofibers is caused by shortening of individual sarcomeres, so segments between the adjacent C lines. The sarcomere shortening is accomplished by sliding of interdigitated myofilaments during asynchronous power strokes of myosin cross-bridges. The relative force generation is dependent upon number of myosin heads forming reversible attachments to the actin filaments. So what mechanisms account for variation in force produced in muscle contraction? Well, engaging varying numbers of motor units. Forces generated depends on the number of fibers and more fundamentally the number of myosin head groups participating in muscle shortening. Alteration in the frequency of stimulation has to do with the summation of force development. And then the length of the muscle prior to the arrival of the stimulus to contract. So length tension relationship. So a motor unit is a motor neuron plus all of the muscle fibers in which it forms synapses. In vivo, each fiber is controlled by a single motor neuron, but each neuron can be wired to multiple muscle fibers. When an individual motor neuron is activated, all the fibers it innervates forms a synapse with are stimulated to contract. We have graded contractions, which are varying degrees of force generation, can be produced by variations in the number of motor units activated, controlled by the central nervous system. And then we have innervation ratios, which are fibers per neuron, which vary anatomically consistent with the functional requirements of muscle in question. So more frequent stimulation yields a greater force of contraction. So twitch is a single transient stimulus and relaxation, and then we have the summation of twitches, which is stimuli sufficiently frequent to not allow full relaxation. These are greater contractions. And then we also have tetany, which is the frequency is high enough to produce steady contractions, so no opportunity for relaxation. And then we have types of skeletal muscle fibers. We have our slow oxidative fibers, we have our fast oxidative fibers, and our fast glycolytic fibers. Our fast fibers are high myosin ATPase activity, thus more rapid cross-bridge cycling and high shortening velocity. So we fatigue rapidly here. And then we have our slow fibers, which has low myosin ATPase activity and lower shortening velocity. So we fatigue more slowly here. And then we have our oxidative fibers, which have numerous mitochondria. They have a high capacity of oxidative phosphorylation. ATP production is dependent on blood-borne oxygen and fuel, which contain myoglobin, which increase the rate of oxygen capture in the fiber. And then we have our glycolytic fibers, which have few mitochondria but a high concentration of glycolytic enzymes and glycogen, which is a polymer of glucose stored as a fuel reserve in skeletal muscles and liver. They are larger and have more thick and thin filaments and therefore can develop more tension, so we fatigue rapidly here. Effects of exercise on skeletal muscle increase contractile activity, so exercise increases which results in the size, hypertrophy of muscle fibers, the numbers of myofilaments, and the capacity for ATP production. Low intensity exercises affects oxidative fibers, so this increases the numbers of the mitochondria and capillaries. High intensity exercise affects glycolytic fibers. This increases the diameter by synthesis of actin and myosin filament and increases the production of glycolytic enzymes. Fiber types may be able to interconvert, and then fatigue is associated with the energy, ATP depletion, loss of efficiency, and calcium storage and release. Muscle is a major consumer of chemical energy and metabolites to accomplish synthesis of myofilaments proteins, so the building of the contractile machinery, the pumping of sodium, calcium, and potassium and other ions, and preparing to respond to the neuromotor stimuli, and then the cross-bridge recycling, so sliding filaments, myosin, ATPase. And then our last lecture, again, is still on muscle physiology. So we have the effects of inactivity and muscle gain versus muscle loss. A classic example is homeostasis. So we have a sedentary pattern of activity, the consequences of aging, disease, disability, space travel, and lifestyle. So which leads to atrophy, which is muscle wasting or loss. It results from inactivity, disease, and aging. Mechanisms of exercise induce changes in skeletal muscle. So muscle fibers do not typically proliferate. A net loss of muscle fibers generally occurs with age. Muscle has some capacity for fiber repair and regeneration. Muscle becomes more massive and capable of increased force generation by individual fibers becoming thicker owing to synthesis of additional myofibrils. Exercise, so mechanical stresses on the tendons and other structures activates genes that cause the muscle fibers to produce more myofilament proteins, so myosin and actin. And then additional nuclei are needed to support protein expression, maintaining optimal ratio of cell volume to nuclei number. New nuclei are donated by the progenitor satellite cells that proliferate in response to mechanical stress. And then microscopic tears in muscle fibers may generate signals that attract satellite cells to repair the breach. Skeletal muscle atrophy. So skeletal muscle has a high degree of plasticity, which is able to adapt in size and composition to needs and activity of an animal. Muscle represents a major reservoir of body protein from which amino acids can be mobilized in periods of disuse. Both reduced protein synthesis and enhanced skeletal muscle Proteolysis provides organisms with free amino acids. So when transient, this adaptation is beneficial. So amino acids can be used to maintain protein synthesis in vital organs such as the heart, lungs, liver, or brain. Amino acids can provide energy by direct oxidation or gluconeogenesis. When sustained, muscle wasting is highly detrimental. So ventilation of lungs impaired when respiratory muscles atrophy, ability of organisms to recover from stress or pathologies is rapidly impaired, increased morbidity and mortality. Muscle atrophy may result from either depressed protein synthesis, enhanced proteolysis, or both. And then he got into many examples of how, so he just went over many examples of all these things. But I'm not going over that with you because there were so many examples. He just wanted to show us mechanisms and how things affect these mechanisms of when our muscles contract and when they would not contract, and what happens when that happens. But anyways, those were our things for today. I also had lab where we learned a bunch of stuff. We dissected today. We learned the shoulder muscles. And yeah. So I love you guys. I hope you guys had a good day today. I still have to go run, so I'm trying to get back into that because I gained a few pounds over break, so I have to get back to where I was, hopefully. But yeah, I love you guys, and I hope you guys have a good day.